Hello, all. Thanks for joining me. It's a Friday night, and what better way to spend it than finishing up our data science project? I worked earlier, and then I did my chores and I ate some food. I started drinking my beer. Yeah, and so it comes in handy when we do stuff that's really boring, and we're at that part where it is quite boring. Because right now it's really interesting because we get to explore the chromosome and see what genes are overlapping these gene bank accession numbers. But it's kind of tedious because it took us an hour earlier just to do 9 of those gene bank accession IDs to get the genes there. And that's how we use our top genes and see which ones can predict the type of autism that we're dealing with, which is language mild, and also savant, which was really the main reason for doing it. We're going to do a 4-class model. So far, we've predicted really great accuracy with just our sets on a 2-class model of healthy as 1 class, and then the type of autism they have, which is mild or language or savant. So all of them scored better than 70%. We can just review it. I think even some of them did score almost 90%, maybe even, yeah, I think like 90%. So those genes are pretty good genes. We used fold change values for that. Oh, Mr. Dallas. Love you, Dallas. Okay. And then let's share our screen. We're just download the link. It is I'm gonna share my screen, the entire screen. And you're welcome to join along. I'm just tracking what we're doing. And we're almost done with this one. There's more work to do. We can look back, scrolling through. This is the document. It's not the actual studio, but you will see when we look back at the percentages of the machine learning that we did, these genes. When we get to the control, okay, yeah, so this one, and then when it predicted, you can just see this from the internet. Yeah, so 71% accuracy for some, 67%. That was a 2-class model. So that was on the testing set. So it generalized okay. It didn't really what is that word called? Overfit the data too much. The healthies, you can see 96% accuracy, 87% on the training set. But then the testing set was a little bit less accurate. We got, it was a combined 12 samples, and there were 8 out of 12 predicted correctly. Okay, so it was almost 70%, but most of them, the savant, were pretty good, 71%. And then when we go to the language, so these genes are good, but they're not really genes. They're gene bank accession IDs, which is basically just the chromosome location. They would never have even been genes if people didn't realize that those were the same locations, or they shared the same sequencing on the chromosome as other pathologies when they're sequencing them. When they run them through their own array or high throughput bead sequencing to match up those nucleotide sequences that make up genes. And I explained this earlier on another one. I could keep reviewing it every time, but I assume you already know what it is. So every one of our cells is it's recycled. It's replicated through mitosis. There's like 4 stages. And then they form some chromosomes. The father and the mother get together. There's a centromere. There's a P arm, a Q arm. These chromosomes, 23 of them. 22 are autosomes. 1 is the sex chromosome. It's either X if you're a female, Y if you're a male. All of them have centromeres. Okay, so when you deal with DNA, those chromosomes are tightly knit in a lot of sequences of 4 nucleotides. DNA has thymine, adenine, cytosine, guanine. So if you see a T in the sequencing, it's complementary DNA. Complementary DNA, it's a double helix, and they bind opposite A to T, C to G, and it just wraps around like this. But then see, it's a wrapped up DNA, right? But it's also wrapping around little beads, like fist wrapping, right? And over little other beads and little beads. And so, and then it tightly knits around the chromosome. Chromosome. Okay, so it's like all of them. All your DNA fits on all of these 23 chromosomes. And there's certain spaces, those histones, non-coding regions. So we got introns and extrons. Whichever one sounds like the one you keep is probably the one you drop, from what I remember, but you can Google it. That's pre-messenger RNA. And what that is really is it's whenever they copy the DNA, the complementary DNA, what happens is it's unfolding, and that pre-messenger RNA is copying that complementary DNA, and it gets wrapped right back around in the DNA, and that is the pre-messenger DNA. Now, whenever it is copied, it's copied by RNA, which doesn't have thymine, it has uracil. So if you see a U, it's going to be the RNA. So you'll have messenger RNA, but really it's pre-messenger RNA. Messenger RNA is made with the amino acids already, the codons, and it goes through a ribosome, which are proteins inside the cell. DNA is in the nucleus. Transcription happens there. Transcription is transcribing off the complementary DNA with the pre-messenger RNA. The pre-messenger RNA has introns and extrons, those non-coding regions. Drop some of them, keep some of them. That's where you get copy number variants, because some of those don't copy the same, but they're the same gene and they still work. The reason you have gene sequencing is because in our bodies, certain genes are getting copied more in certain pathologies or in certain, superhuman qualities than other typical folk. But we all pretty much have the same genes. And then they're made into proteins. Now, proteins are usually single-strand protein, like insulin. It's just a single strand. You just make them, and you can have a lot of them. But a lot of the proteins there are wound up. So you can have primary, which is like the single protein string. That's after the messenger RNA goes through the ribosome, and it makes those amino acids. It makes the amino acids that are the protein strings. And from there, you can have secondary, which is like the alpha and beta pleated sheets. You could have tertiary, which starts doing the looping. So pleated sheets, for some reason the proteins get together, or the alpha sheets. And then you have the tertiary, it starts winding. And then you have the quaternary, which starts winding and mixed with another protein, like hemoglobin, quaternary, so that it can hold in certain other enzymes or proteins like the hemoglobin. Certain copy number variants are seen in, they like to give the example of sickle cell anemia. And sickle cell anemia is usually like the replacement of valine with a different amino acid. And that was something that happened in evolution whenever the parents had malaria and then they gave birth. And then their child, through meiosis, tried to defend against malaria again. And it prevented it by the shape of the blood cell. And it became shape of a crescent, a moon, half moon, quarter moon, or whatever. So they call it a sickle cell. But the thing is, they don't last very long because they can't hold that much oxygen, and they have a lot of complications, immune dysfunctions, and stuff like that. Without intervention, they need to be monitored all the time. And here we are on autism spectrum disorder. So when we get these genes, when we find them analytically through big matrices of these gene regions that it's processing, there's sometimes different ways that this information is stored. Like these were MEV files. Sometimes you get a simple CSV file and that's fine and all the data is already there, but it already went through the processing, so you don't really get to scrutinize it. And we're not going to scrutinize it anyways. We're going to assume that it is there because we're using the quality score count. And that's how we actually merged all these samples. We're just using that. So we had to build this data. Then we had to filter it. And then we had to test it and analyze it. And then we use those genes from fold change as top genes, because in any pathology of cancer or organ dysfunction, you're going to have more genes in that region, in that tissue region, that will be producing more, and there will be more dysfunctions. Most of the times it's immune dysfunctions, like tumor cell regulators. They're supposed to keep bad cells from replicating, and these are pretty much retarded by some pathology that has come in there, or stress or immune dysfunction, just like a retarding vector. Okay. It retards the normal function. It quiets things down. It creates this dysregulation of the immune system. And like viral infections and radiation damage, usually our DNA can replicate and heal itself with little repairs, but then sometimes it's too much, and so they just allow the cell to be killed. And if it reached any other cell, they kill those cells in a healthy immune system. But sometimes that doesn't happen. Stress takes over. The body doesn't really want to do anything really other than vital functions, and the rest of the body is just like, like just tearing down itself, letting whatever happens because the immune system is just not up to date because of stress taking over, not getting enough sleep, poisoning the body with toxic waste products and inflammation from the diet, stress, free radicals, other sorts of things that your body needs, like rest to heal, hydration, exercise to get things flowing, not staying stuck in one spot, stuff like that. Good stuff that you need to do but aren't doing because you're focused on one thing that's really tearing apart your body. So a lot of students can actually get sick, and they're otherwise healthy if they didn't stress themselves out that much. As one example, also people that are working, 12, 15 hours a day, stressed out the whole time, just struggling to make ends meet. Those types of people. Other people like cancer, it's from lifestyle changes or genetic mutations. Sometimes they just inherit it. There can be their telomeres are bad. It doesn't function right. They could have, just had some kind of viral infection and it took over, because even the emotional stress can be stressful. Sometimes people have too much emotional stress, and that's too much for their body. That's why you see people that you're like, why does it have to be this person? And then you're like, I don't know. Well, you know what? It's probably because they're stressing themselves out too much and you don't see it, or they don't see it. But it also is, most of it's genetic. They say it's genetic. So the diet, genetic, whatever's going on. But yeah, a lot of it is genetic. That's the gene, epigenetic. It's like, gene centered around genes, gene expression, the whole genome, all the chromosomes. Now, if you talk about mitochondrial DNA, that's the mitochondria, which is an organelle. You inherit the organelles from your mother. Whenever it goes into meiosis, the ovum, the organelles are in there, matches with the spermatocyte from the male. That's where you combine the DNA, but you keep the organelles from the ovum from your mom, and that includes the mitochondria. Mitochondria has DNA of itself from your ancestry, and then you inherit the ancestry DNA. And that's what they use to test your ancestry. And if you know for sure you're mixed, and let's say your dad is white and you've got like green blue eyes, and nobody on your mom's side who's like a person of color has it, and then you get a DNA test and it says, oh, you're all color. Well, it's not because it's wrong, and it's not because you're not, mixed. It's because it's mitochondrial DNA. And everybody on your mom's side was of color. And in reverse, it could actually work the same too. Someone of color gets a test, and their mom's white, but their dad's a color, of color. Then they're not of color. They're all white. Well, mom's DNA, and that person's obviously not white. So phenotype. So anyways, let's see how well the language did on prediction. Are we just giving you a little rundown? And then why do we do this? Why do we find these genes? Well, when you find these genes, if they're top genes, if they're lit, they're going to predict pathology when you see it in a lot of the population. Many samples will have it. Some samples won't. That's why it throws it off. So you have to try and get as close as you can to the sample as possible. And then you get these genes. These genes, they basically can replicate them with machinery. They can take that messenger RNA. They know what it is. They can take out the bad copy variant, replace it literally, and then duplicate it in vitro. They have different substances they know take out certain nucleotides and amino acids, or it can break apart and bind them, give them their shape. And they use there's proteins that give it its shape. So it's not like they're looking at it. They do look at it, histologically and see the shape, but you have to see it at a fine microscopic level. But you can also flag it with genes that they know are genes that make the shape, which are like transcription factors. Also, microRNA, that stuff will actually, with microRNAs, miRNA, those are actually things that can inhibit proteins from being made, or they can stimulate them from being made. And they don't just work on one gene protein. They can work on multiple proteins. So they use microRNAs. I learned this from a different study. It was the one on actually mononucleosis. Yeah, the one that actually Epstein-Barr virus is for sure linked to. Mononucleosis, the study that I looked at, had microRNA, and none of my other study uses microRNA, so I couldn't really use it, and it doesn't show up as DNA. Those are basically like helper proteins similar to ribosomes, but they control which messenger RNA is going to get made. They basically blacklist the ones that won't, and then they promote the ones that will. Now they can take those microRNAs, create them in a lab, put them in with other with the cells. When you get stem cells, that is gene therapy stem cells. You got protein-rich plasma, which is like fibroblast sports athletes get whenever they split their menisci and they need to get into the game soon, or their labrum and they need to heal. They inject it in there, and it's like basically all the supplies to building a home in their labrum is there. So you don't have to keep moving the arm, waiting for synovial fluid to get in there. You do have to do physical therapy, but it's already there. That's why they heal faster, and it seems insane how fast they heal. And then you have the genes that go with stem cells. So stem cells, those are basically using, you've heard of the cord blood. It's stem cells. So unless it's pluripotent stem cells, which you have to get from an embryo, and because of ethical reasons, when people do in vitro fertilization, they make these embryos and they're ready to be implanted. Now, if they're between 2 days old and no more than 7 days old, 2 to 7 day old embryo, which is just a fertilized ovum that has divided into its blastocyst form for up to 7 days, no more than 7 days, those stem cells can make any cell in the body. That's why you have some people that have ovarian cysts, and because of the way that it divides in the ovary where the ovum are, it's similarly making pluripotent cells in their ovary where you'll have a cyst in the ovary. We've seen this in textbooks in science, where there's teeth, hair, looks like different tissue types of many things in there, and it's just a cyst on the ovary. Because the pluripotent stem cell can be charged. It can be made. This is a fascinating part of genetics. If you've ever taken genetics, it can be made scientifically in a lab to recreate any tissue type. It can make heart tissue, cardiac tissue. It can make liver, hepatic tissue. It can make spleen tissue. It can make brain tissue. It could literally make anything. Bone tissue, fingernails, skin. So you have pluripotent stem cells. Like imagine that for a fire victim with their skin burnt off, wanting their own skin. Well, you got pluripotent stem cells and you have the genes there that you want. You take your own skin, you extract the genes for the skin with that DNA, you make tons and tons of proteins out of that. You put it in a pluripotent stem cell in vitro, you do the whole cooking process, this temperature, this rotation, this days, this times on a timer. You can grow the skin and literally plant it on a burn victim. Yeah, and there's a whole process for that. You probably look that on YouTube, but I mean, that's what it is. Okay, now, so if you were to make the other tissue, and they've done that, I mean, they've been using it actually, similarly when they grow it in labs for all that lab-grown meat, they've been doing stuff for decades, actually. The first one was a sheep that they reproduced from a cell, and then there was something in Japan where they did the same thing with the CRISPR with mice, and they found out that after 57, I just read about this article, and it wasn't a peer-reviewed article, it was just like a science one, just like, okay. 57 generations of the mice were perfect, they were fine. Suddenly, all of a sudden, the 58th generation started breaking down. They died. All of them died within 4 days. So they're like, that's weird. So they did it again, and the same thing happened. So for some reason, you can replicate that 57 times. And it might have something to do with the telomeres. They brought up the telomeres when they were doing this HeLa cells experiment with Henrietta Lacks and how everybody's vaccinated with HeLa cells, which is basically HPV. So they used hers because it can replicate faster, but it saved a bunch of animals from being tested because they were using monkeys before that time to grow in a slow amount of time vaccines to replicate genes. So they have a fast way of doing it. And 90% of the population has Epstein-Barr virus. Some of the studies I've seen use Epstein-Barr virus, similar to the HeLa cells, which are HPV virus. But they've been retarded. They don't grow. They can't replicate themselves. They're as slaves to replicate themselves, but we've attached our genes to it, so they grow our genes. Now, in other situations where we're stressed out, they attack our genes and take our genes and mutate them. So think about that. But also, whenever you have these genes, they can replicate these genes. They can put them in, make vaccines, make tissue that you need to repair. They can treat cancers of all sorts. If you have like a disease of an organ, they can use that. They can lab grow it. If you donate your embryos, who's to say if they're going to be born children or if they're just going to use your embryos to grow tissue for somebody else, or just to destroy it after growing it because of ethical reasons, allows you to do something weird like that, but not to give it to somebody else to use. And I think there is some ethical tape around that. So you can't actually use the embryo to replicate and grow tissue. So there was a guy in Japan with the CRISPR machine, and he said he experimented on people, which he wasn't allowed to do because it was outlawed worldwide. It's outlawed. I think it was Japan. It might have been China. I'm not sure. Sorry. Okay. So anyways, everybody knows about it. It was like 2 years before COVID broke out, and it was around the same region, similar lab. So they're like, oh, maybe they were trying to do something. Or like, ooh, let's make something. So that was a big thing. So people didn't like Chinese people for a while. But this guy, he made babies for parents that had HIV, and he claimed that the babies did not have HIV because he used the CRISPR machine. He did everything right. He was a one-person lab. He didn't have a consortium to deal with. He did it illegally. He made lots of money. He also went to prison. He's probably still in there, or else they killed him. I'm not sure. Or he's working in a lab for free, making crazy stuff for them. Who knows? Because COVID did come out 2 years, but I'm not saying that's what happened. Who knows what happened? Because they are communists. Seriously, they're still considered communists. I'm part Asian, but my mom's Filipino, and I don't have a problem with Chinese people, but I don't know how their government works. But I would never want to get in trouble or break a law in China because they're known to be pretty harsh. You'll probably die in one of the Chinese jails. Same with Korean jails, and suffer horribly before dying. And then we are going to get into it. This is the perfect time to drink beer when you're doing data science, is whenever you're doing something really boring like what we're about to do. But here we go. So you have a little, and you know what? It turns out that guy, they found out years later, the kids actually ended up having HIV anyway. So he didn't do it right. But there's people that do it right. So it takes a team. Because you can't possibly be there that whole time doing everything. Like if you read these research articles of all the things they have to do, it is a timed process. You have to do it exactly this way to get the same results, or to go through the gene expression profiling machine accurately. Yeah. So he was doing it himself, or else he hired a lazy team, or someone fell asleep. That happens. Didn't get done right. And they lied and said, hey, it was. And he's like, they said it was. They never make mistakes. They're A+++ people. And they lied, and they actually just took their money. And so they're always doing that. Okay, so we're let's go ahead and open up our. Oh yeah, we were gonna look at that, huh? Let's we can look it up. Hold on real quick. We're gonna go into the actual document, and it is right in we're not doing that. I do not take advantage of AI. It's just Copilot. I asked Google, and I have a Microsoft internet on. So that's as far as I use AI. I do not have it use anything else. Okay, so we're going to go into this folder. It's just this R document. You have the link. You can just run all chunks. You could have ran all chunks right when I was talking, and then you'll be where we're at. We're not really running any code right now because we're still building this list. We're exploring. We've done these many gene accession IDs, and then found along a search of the chromosome what gene overlaps that nucleotide sequence, and got that. This one has 2. I think I'm just going to make another row for that gene, because there's 2 genes that overlap this location of the chromosome. So I'll just make another row. So we're going to have 48 genes. We'll probably have more. Okay, so we're doing this one now. Let's just look at the, we're going to go ahead and look up how well our functions did. Okay, so that was the last one. This one did. Okay, that was the one we looked at, and it was 71%. It was about 70% accurate. And then let's go to another one, the language one. That one was 75% accurate. Now let's go to another one. The mild one was 82% accurate. So the mild one was the best. These genes for the mild are really good. Then comes the language. That one was 75%. And then comes the savant, which is 67% accurate. So that's good, because you look at my last model, a lot of them weren't doing that great because we couldn't use fold change for that. Fold change is actually pretty powerful. They use many different ways to get the top genes. Most of them use differential expression, or they use the PCA, and look at the ones that are having the most variation from a line, where the line is figuratively where if you follow that formula, every sample will fall on that. But because there is no strict formula, there's a lot of splatter, a lot of spots, samples outside of that line, figuratively, in 3D space, 2D space, 3D space. All right, so anyways, without getting too technical, we are going to go finish this. All right. It's always good to have water too. It's refreshing. Okay, so we're just going to go through. Control C in my PC. We're going to go to the NCBI gene site. We can just Google it. We're going to get the FASTA. Once we Control V paste this, it's going to show us the FASTA. Here's the link. Or we can just go to BLAST. We'll just go to BLAST. There you go. Quick and easy. Now we're going to make sure that this is selected, genomic and transcript database. And open in a new window, highly similar. And we're going to basic local aligned search tool, blast it. We just wait, and then we're going to take the first highest ranked genome that it finds. This one has one right here. Sometimes we will find the same chromosome location. Okay, we're going to go to graphics. We're going to look at the chromosome right here. I brought my glasses too, because you know, sometimes my eyes just get lazy. Because I'm old. And we have tiny little muscles that open and close our lens to diffract and extract light. Oh, this is great. I can see. Wow. Okay, so here we are. This is the gene access code, gene bank accession code we were looking at. We have one gene that's overlapping. Here we are in this window. That's that little gene right there. We can zoom out and see if there's any other. It's 12. It doesn't really say the location, does it? Of the okay, so we shrunk this down pretty low. So it's not really. So it's not really it's not saying P or Q for the short arm or the long arm. Short arm is above the centromere of the chromosome, and Q is below the centromere of the chromosome. So what we have is this gene, RAP1B. Okay, it's an oncogene. RAP1B. Okay, so that is for our this one. RAP1B. We're building our vector as well. Quotation marks here. And then we're going to go ahead and look up this one. Oh, it's kind of dizzy whenever I don't. Whoa, when I don't really need it. Yeah, I'm okay with squinting. That's fine. Yeah. So I'm going to go ahead and do that. Right here, we can close out of this window. There's only one gene there. We can close out of this window, and we're just going to go right in here and Control V. This gene bank accession, everything's checked, and then search. Oh, nothing was found. Let's click Y. Oh, okay. A1348022. Oh, it's AI 348022. Nothing found. Oh, that sucks. You know what? Maybe we can just go back to the FASTA sequence and use that one instead. Gene, we're just going to enter that in. Look at the FASTA. Just copy this. Copy it. And then we're going to go back over here. Control V. And then search for it. Oh, yeah, so nothing was found. Okay, let's just see what it found then on the graphics, because I guess we can look straight on the graphics for it. Yeah, it's not really showing anything. Gene bank. Let's look at gene bank. All right. Sample. Huh, this is weird. Okay, so we're probably gonna have to drop that one. Citation. There's not really. Experimental databases? Where are eukaryotes? Prokaryotes are, I think, are the plants. Virus, or it could be bacteria. Oh yeah, because eukaryotes are plants and animals. I think. Exactly. All right, so we're just gonna have to leave that one. All right, nothing. NA. We got an NA. All right, for this one, now we're gonna go here. And we are going to go back to this one. Control A, delete, Control V. There we go. Should really switch to a PC. There we go, chromosome 1. Let's go to the graphics. It did rain lightly today. I didn't think it would. Oh. Wow, here we are. And here this is. But why does it look like there's nothing overlapping this window, but there are genes here? Okay, it looks like we got 3 genes overlapping. There's a non-coding region, I think it is. A transcript. messenger RNA, and then this gene, TLCD4. And this is our query right here. That was our query. So it looks like our gene is, let's just zoom back in again, just to be sure. It's this gene, TLCD4. TLCD4. And then we'll go to the next one. Next one here. Control C. And close out of this. And this. And just go here. Control V. Everything's checked. I think I'm gonna put my glasses back on. Oh, goodness. Just for reading. Okay. Let's scroll down. We're on chromosome 9. We're going to go to the graphics. Here we are. And then we got a gene right here. It's RIC1. RIC1. Okay. 1. And then we'll add it to our vector above. RIC1. There it goes jumping again. Let's cut this. And we'll put it right here. So we can see as we work. All right. The next one, we'll go ahead and Control C. And then we're going to use our window. We can remove this and that. And then we're going to go up here. Control V. Everything's checked. That should be same window. Oh. Okay. Chromosome 2. Let's see. And then we have the graphics. Here we are. That's our query. And then here's a gene. It's on the forward strand. ATF2, activating transcription factor. Okay, ATF2. ATF2, okay. Let's go and put it in our vector. And then our next one, we'll go ahead and copy that. Get out of this window. Control V. Blast. Chromosome 7. Going to the graphics. Here we are. One gene. It's on the forward strand. KBTBD2. KBTBD2. And that stands for the stop sharing thing is in the way. I had to move it. Kelch repeat and BTB domain containing 2. Kelch repeat. KB TB D2. 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 Okay. Let's close this. I'm going to go to our next one. Okay, we got one. Chromosome 3. The graphics. Here we are. That's our query. And no track data found in this range. Oh, well, that's a bummer. Here we are right here. This is the window we're looking at. But it looks like there is a gene right there. Let's go ahead and zoom out. See how far out that gene is from us. This is us now. And there is no gene here yet. Wow. Okay, let's see how far away it is. There we go. The closest one is this one. It's in the 156 million range. Ours is in the 156 million range too. So we can say that this gene is probably it. We'll just take that gene. TIPARP. Okay. TI PARP. TIPARP. All right. TIPARP. TIPARP. Okay. Now we'll go ahead and look for our next one. Oh, we didn't put a comment in this. All right. Let's go ahead and do that. We're going to copy this one, Control C. Actually, if you use an Apple, you probably are better off with an Apple. I have an Apple iPad and I'm just used to a PC. So whatever you do to copy or paste, I'm sure it's fine, but it's not going to be the same thing as what I'm doing in a PC. Okay, let's search. All right, we got one, chromosome 11. Let's going to do that. Oh my gosh, this chair. It's giving me like muscle aches. Okay, so here we are. That's our search, and we do have a gene here. It is TUT1. UT1. I'll go ahead and put that there. And then we'll copy our next gene and look for that one. Okay. Chromosome and the graphics. Here we are. Now it looks like there is a gene there, but there's also another gene bank accession ID. So we will not worry about that one. Because it's just another gene bank accession ID. Then I'll just find this gene again. It's good if I want to compare in other studies where there are some things that compare it. People look up different sources by another method, PICO, when they're just looking at different studies that are presented and they look for keywords in that study. That's when that becomes important, the gene bank accession ID. And they filter until they get what they want. Okay, so it looks like Myo 1B. We learned that in chiropractic school, but I wasn't really interested in it. I didn't really want interest that one. You can't really do anything with that. They don't like math majors in that profession. I think you're crazy. I have no respect for people that think I'm crazy just because I majored in math. Okay. Myo 1B. All right. Now let's go on to our next one. Control-C copy. Close this window out. We're going to go over here. Control-V. Paste. And chromosome 12. We're going to go to the graphics. Here we are. And there's a gene. Look at that. It's on the reverse strand. WNK1. And it looks like there's only one. So we'll put that WNK1. WNK1. Okay. And then we'll go for the, let's control S this because we're not going to save it otherwise. All right. Control C copy the next gene. Close out these windows. Control V paste. I got two. We're going for the top ranked one. They're actually very similar. but it's the top one. And so we'll just do this one. Go to the graphics, comes on bottom. Here we have one gene. Also, here we are. That's our query for the gene bank accession ID. And then this one, sting one. Stimulator of interferon response. Cyclical is that little c. Adeno monophosphate. Guanine, adenine monophosphate. I remember some things. Okay. Guanine. So that's cyclical GAMP, interactive 1. Sting 1. Put a comma there. Okay, we'll go on to our next one. We'll Control-C copy that. And then we'll close these windows out. And we'll go here and Control-E paste. And these check that should be. And chromosome 1. And then we'll go to the graphics. Stop sharing. Not yet. Okay, so here we are, and there's a gene here. It's CFAP144, cilia and flagella associated protein 144. There's cilia in your duodenum, in your small intestines. They take nutrients into your body. That's where we digest all the minerals and nutrients we need. And then it gets taken to the large intestine. The flagella. Those I can only think of in the reproductive system. But I guess the cilia, yeah, I don't really know of any other place. So gene cards would know what that is, but CFAP144, if you want more information. CFAP144. Let me make sure that's right. CFAP144. Okay. Yeah, it is bring your own beer. So if you have beer, you should drink it. Wine makes me loopy, so I can't work when I drink wine because I want to make fun of everybody and I want to laugh at them, their expense, and then I just can't do any work because I'll just start laughing and being mean. Okay, and that's just so funny to me when I'm drinking wine. But I can drink beer and do this, it's fine. So bring your own beer. Let's move this down. So we can keep seeing it. All right, so we're gonna go with the next one. I'm not sure how many we did, but some time has elapsed. Let's see. I think we did about 10, and it has been an hour. If you need a break, go ahead and take one. I'm going to do this one, and then we'll take a break. Yes, a 2-minute break. You're only allowed 2 minutes. That's it. Okay. Let's go ahead and copy this one. And then we'll go ahead and close this window out, close this window out, and then we'll Control-V, search for it. Oh, man. Crohn's too. This bar stool is really like it's giving me like almost sciatica. Graphics. Here we are. And then there is one gene. See, chimerin. That's like the chima in the gallbladder. What is that? The cisternochyli chimerin. Chimerin. Chitin. Chitin is like the hard shell of arthropods. I think they're exomorphs. I'm not sure. What do you call it? Insects. It's been all since biology. CHN1. Wow, let's just copy it. We don't have to remember if we just copy it. Okay. Just make sure that it pastes the right thing, though. All right. And we'll go to our next one. So I go ahead and take our little bathroom break. Control S. Save. Oh, God. Oh. All right. Oh, we're back. Stretch. Okay, you should be back from your break now. It's Friday night. We're having so much fun doing this data science work. Bring your own beer. Okay, let's go ahead and copy this next one. Well, this is fun. I think it's super fun. All right, so I'm going to go ahead and close this. And then we're going to search for it. Okay. We'll go to the graphics. Here we are. We got a gene on, one gene on the forward strand. Atom, TS5. Let's just copy that. Atom, metallopeptidase with thrombospondin type 1, motif 5. The motif sound familiar. I believe that's something that has to do with the introns, extrons of pre-messenger RNA. Oh, it's been a while. We don't really need to know that. It'll explain what that gene is. Okie doke. All right. And then our next one, I'm going to copy. What else? Go ahead and set it for the next one too. Okay. So we're gonna just copy this next gene bank accession ID. Close out these windows. And search. This is my last beer in the house. You're allowed 1 to 2 per night. So we have another NA. It is fetal liver spleen. I feel like we've seen that one before. Okay, so we don't have anything with this one. We have an NA. We're gonna have to put NA right here. It's our placeholder. Right, that's our second one. And we'll control the same. Let's look for our next one. All right, we got one. Chromosome 20. I don't think we've had any that high yet. Okay, here we are. Another gene bank accession ID. We're gonna look right here. Unless somebody else is doing the same work at the same time. We're not really sure. So there's that Saurus fetal liver thing again. But this one is our CST7. Copy that. Oh, it didn't copy it. That's not cool. Hold on, we have to go back in there. Okay, CST7. Copy. Now we'll go back over here and paste it. Control S. Let's go ahead and copy our next one. Close these windows out. Probably gonna start standing pretty soon. I have to stand up this one. Oh my gosh. Okay. All right, I'm gonna go with the top ranked one, 96.10%, chromosome 9. To the graphics. Here we are. And last results. We got one on the forward strand. It's AQP7, aquaporin 7. I've seen it before, but I don't really recall what it is. Not 7. I think it's just aquaporin in general. Control S. We got one here. Let's copy that. Go ahead and go to our last site. Control V, paste. Blast. I'm standing. I think this wood bar still. That's fine. I'm not doing a lot of typing anyways. Nothing found here. We got another NA. So we have to put in A as a placeholder. And we'll do our next one. Let's copy this. Control C. close this window. Control V. So these ones that are AI, they don't seem to have anything. We're going to have quite a bit of NAs in, since a lot of them start with it. I'm get another NA. Copy this one. Close this window. Control V paste. And we have a chromosome graphics. This is our search query. And